Bacteria are everywhere. These microscopic organisms inhabit virtually every corner of our planet, from the deepest oceans to the highest mountains, and even within our own bodies. Though invisible to the naked eye, bacteria play a crucial role in shaping our world, driving essential ecological processes and influencing human health in both beneficial and detrimental ways. To understand the vast diversity and ecological significance of bacteria, scientists have developed various classification systems based on their physical characteristics, metabolic processes, and genetic makeup. These classification systems help us identify different types of bacteria, understand their roles in the environment, and develop strategies for managing both beneficial and harmful interactions with these microscopic powerhouses. One fundamental aspect of bacterial classification is their morphology, which encompasses their shape, size, and arrangement. By examining these physical characteristics under a microscope, we can group bacteria into broad categories that provide valuable clues about their potential functions and behaviors. In the following sections, we will delve into the fascinating world of bacterial classification, exploring the diverse shapes and sizes they exhibit, their preferences for specific temperatures and pH levels, their relationships with oxygen, and the crucial role of Gram staining in distinguishing between different bacterial types. Bacteria exhibit a remarkable diversity of shapes, a testament to their adaptability and evolutionary success. These shapes, often used as primary criteria for bacterial classification, are not merely aesthetic variations, but reflect underlying structural and functional adaptations that allow bacteria to thrive in diverse environments. The most common bacterial shapes are Kochia, Bacilli, and Spirilla. Kochia are spherical bacteria, resembling tiny balls under the microscope. They can exist singly or form characteristic arrangements such as pairs, chains, or clusters. Examples of Kochia include Streptococcus pneumoniae, a common cause of pneumonia, and Staphylococcus aureus, responsible for various skin infections. Bacilli, on the other hand, are rod-shaped bacteria appearing as elongated cylinders. They are often found singly, but can also form chains, like the bacterium Bacillus anthracis, which causes anthrax. Spirilla, the least common of the three major shapes, are spiral-shaped bacteria resembling corkscrews. Helicobacter pylori, the bacterium responsible for stomach ulcers, is a well-known example of a spirillum. Bacteria, like all living organisms, have optimal temperature ranges for growth and survival. Based on their temperature preferences, bacteria are classified into distinct groups: psychrophiles, mesophiles, thermophiles, and hyperthermophiles. This classification reflects the remarkable adaptability of bacteria, allowing them to colonize environments ranging from icy glaciers to boiling hot springs. Psychrophiles, as their name suggests, thrive in extremely cold environments with optimal growth temperatures below 20 degrees Celsius. These cold-loving bacteria are commonly found in polar regions, glaciers, and deep oceans. Their unique adaptations, such as enzymes that function optimally at low temperatures and cell membranes with a higher content of unsaturated fatty acids to maintain fluidity, allow them to thrive where most other life forms struggle. Mesophiles, on the other hand, prefer moderate temperatures with optimal growth between 20 degrees and 45 degrees Celsius. This category encompasses the majority of bacteria, including those found in soil, water, and the human body. As we are mesophiles ourselves, it's no surprise that many bacteria that inhabit our bodies share this temperature preference. Beyond temperature, the acidity or alkalinity of an environment, measured by its pH, also plays a crucial role in shaping bacterial communities. Like their temperature preferences, bacteria exhibit a wide range of pH tolerances, reflecting their remarkable adaptability to diverse habitats, from acidic volcanic soils to alkaline soda lakes. Based on their pH preferences, bacteria are classified as acidophiles, neutrophiles, and alkaliphiles. Acidophiles, as their name implies, thrive in acidic environments with pH values below 5.5. These acid-loving bacteria are commonly found in environments like volcanic soils, acidic hot springs, and even the human stomach, where the acidic environment aids digestion. Their adaptations include specialized cell membranes and enzymes that function optimally at low pH levels. Neutrophiles, the largest group of bacteria, prefer neutral pH environments, thriving between pH 5.5 and 8.5. This category encompasses most bacteria found in soil, water, and the human body. Our own bodies, with a generally neutral pH, provide a suitable environment for these bacteria. Oxygen, essential for the survival of many organisms, plays a complex role in the lives of bacteria. Some bacteria require oxygen for their metabolic processes, while others are poisoned by it. Based on their relationships with oxygen, bacteria are classified as aerobes, anaerobes, facultative anaerobes, and microaerophiles. Aerobes, like humans, require oxygen for respiration, a process that generates energy from food. These oxygen-dependent bacteria are commonly found in environments where oxygen is readily available, such as soil, water, and the Earth's atmosphere. Their metabolic processes rely on oxygen as the final electron acceptor in cellular respiration. Anaerobes, in stark contrast, cannot tolerate oxygen and may even be killed by it. These oxygen-sensitive bacteria thrive in environments where oxygen is scarce or absent, such as deep sediments, the intestinal tracts of animals, and anaerobic digesters. They obtain energy through fermentation or anaerobic respiration, utilizing alternative electron acceptors like sulfate or nitrate. In the vast and diverse world of microbiology, identifying and classifying bacteria is paramount for understanding their roles in various ecosystems and developing effective strategies for managing their interactions with humans. While microscopy reveals their shapes and arrangements, a technique called Gram staining provides crucial insights into their cell wall structure, a fundamental characteristic that dictates their response to antibiotics and other treatments. Developed by Danish bacteriologist Hans Christian Gram in 1884, Gram staining is a differential staining technique that differentiates bacteria into two major groups, Gram-positive and Gram-negative. This distinction, based on the ability of bacterial cell walls to retain crystal violet dye during the staining process, reflects fundamental differences in cell wall structure, which have profound implications for bacterial identification, treatment, and pathogenesis. Gram staining is a cornerstone of microbiological analysis, providing rapid and valuable information about bacterial identity. This simple yet powerful technique has revolutionized our understanding of bacterial diversity and continues to be an indispensable tool in clinical diagnostics, environmental monitoring, and basic research. The Gram stain procedure, a cornerstone of microbiological analysis, is a straightforward yet elegant technique that differentiates bacteria into Gram-positive and Gram-negative groups based on their cell wall properties. This differential staining method relies on a series of steps involving dyes, mordants, and decolorizers to reveal the structural differences in bacterial cell walls, providing valuable insights for bacterial identification and characterization. The process begins by flooding a heat-fixed bacterial smear with crystal violet, a purple dye that stains all cells purple. This initial staining step ensures that both Gram-positive and Gram-negative bacteria absorb the dye, setting the stage for differentiation based on their ability to retain it. Next, iodine, a mordant, is added to the smear. Iodine forms a complex with crystal violet within the cytoplasm of the bacteria, enhancing the dye's affinity for the cell wall. This complex is more difficult to remove from Gram-positive bacteria due to their thick peptidoglycan layer. The crucial step in Gram staining is decolorization, typically achieved using a mixture of alcohol and acetone. This step dissolves lipids in the outer membrane of Gram-negative bacteria, allowing the crystal violet iodine complex to leach out, effectively decolorizing them. Gram-positive bacteria, with their thick peptidoglycan layer, retain the complex, remaining purple. Finally, a counterstain, usually safranin, a pink dye, is applied to the smear. This step stains the decolorized gram-negative bacteria pink, providing a visual contrast to the purple gram-positive bacteria. The counterstain allows for easy differentiation between the two groups under a microscope. The gram stain, a fundamental technique in microbiology, differentiates bacteria into two major groups, gram-positive and gram-negative. This distinction, based on the ability of bacterial cell walls to retain crystal violet dye during the staining process, reflects fundamental differences in cell wall structure, which have profound implications for bacterial identification, treatment, and pathogenesis. Gram-positive bacteria, stained purple by the gram stain, possess a thick peptidoglycan layer in their cell walls. This robust layer, composed of sugars and amino acids, provides structural integrity and rigidity to the cell, protecting it from osmotic stress. The thick peptidoglycan layer acts as a barrier, retaining the crystal violet iodine complex during the decolorization step, resulting in the characteristic purple color. Gram-negative bacteria, on the other hand, stained pink by the gram stain, have a more complex cell wall structure. While they possess a thin peptidoglycan layer, it is sandwiched between an inner and an outer membrane. The outer membrane, rich in lipopolysaccharides, renders these bacteria more resistant to certain antibiotics and detergents. During the decolorization step, the lipid-rich outer membrane is disrupted, allowing the crystal violet iodine complex to leach out, resulting in the pink counterstain.